Hey, welcome back, everyone. I think we can move on to our next speaker, Wei-Zen Wei, from Wayne State University. Wei-Zen Wei, it's over to you.
Thank you very much. I am happy to discuss our effort to identify germline cancer risk genes that can alter the course of the disease. And we use diversity outbred F1 mice as our discovery platform. So why is this important? Because we know there are oncogenes and tumor suppressor genes that are the major factors in cancer occurrence. But there are germline risk genes that can either impede or accelerate this process, and this is clinically important. However, they have been difficult to identify until we came across diversity outbred mice and discovered that they are very good discovery platforms for our work. So the platform we started out with is BALB mice that express a transforming red gnu, which is a homolog of human HER2, and that is an oncogene amplified in about 20% of breast cancer and cause a more aggressive disease. So in female mice that express the new oncogene, they can develop spontaneous tumors in all 10 mammary glands. When we did the CT scan of the mouse, you can see nodules in every one of the mammary fat pads. And the liver and bladder lights up because they are the route of excretion for the tracers. So we would like to know if Rat gnu continues to drive these tumors throughout the disease progress. So we did an image with PET using a monoclonal antibody that recognized Rat gnu and is conjugated to copper-64. So they light up under PET imaging. And you can see every tumor lights up, showing that they retain the expression of Rat gnu, and that continues to drive the disease progression. And so we want to know if there are genetic elements that would change the occurrence of these tumors. So what we did is that we take these BALB T mice that are maintained as heterozygous, one chromosome express this transforming Rat gnu, and we cross these mice with the diversity outbred. So the F1 mice carries one chromosome from BALB T that express the transforming gnu, and the other chromosome from the diversity outbred parent. So it has a mosaic of 8 different possible genes from the founder strain. So these F1 mice, called BALB by DO F1 UT mice, have 8 possible diplotypes. And compared to the 36 possible diplotypes, it is much smaller variation, but it makes it possible to do the mapping with a smaller number of animals. And so in the BALB T mice, the tumor begins to appear when the mice are about 14 weeks of age, and it spreads over a 5-week window. And in the BALB by DO F1 mice, you see the tumors begin to appear much earlier. So at 9 weeks, they begin to show up, and it spreads over a window of about 14 weeks, so much wider range. So it is clear that different genetic background can alter the course of disease, and tumors appear at different times. So we use tumor onset age and tumor growth rate as our readout to try to look for the QTL that dictates this changed tumor behavior. So we did the straightforward RQTL analysis of tumor onset age. We bred maybe 400 or 500 mice to identify the females that also express new oncogene. And we did this analysis using tumor onset age versus all the markers on the GigaMUGA chip. There are 2 peaks that stand out, one in chromosome 1, the other in chromosome X. And when we look at the haplotype effect in chromosome 1, the peak region that has the highest LOD value also corresponds to an area that has a very divergent onset age. So the earliest onset age, namely the shortest latency period, correspond to the PWK and the Cas haplotype. So if this region is of these two haplotypes, then we can say the tumors may come early. So we look at the same analysis in chromosome X. Again, the peak value corresponds to a region where it's divergent in tumor onset age, and the earliest would be also PWK and Cas. So it is interesting to us that this analysis actually point to these regions that both predict PWK and Cas as the more aggressive tumor. So then we analyze the tumor growth rate using this equation. We determine the tumor volume of every single tumor in the mouse, add them all up, and take the natural log of that value. So now this equation becomes linear, and then over time, there is a straight line, and the slope of that line is what we use as representative of the growth rate. We plot the growth rate and analyze it against the genetic markers on the gigamuga, and the most prominent peak that stands out is in chromosome 10. Interestingly, when we look at the peak value of chromosome 10, the highest LOD value again corresponds to this highly divergent tumor growth rate region. And the highest growth rate, the largest value, the steepest slope, also correspond to PWK and Cas. So we were surprised by how really this prediction comes together, that PWK and Cas predict the most aggressive development of memory tumor that is actually driven by HER2. So there is a very strong element of genetics that can alter the course of the disease. And we wanted to verify that by testing the hypothesis that if we put PWK genetic background into the BALB new T mice, it will change the course of the disease. And so we generate these F1 new T mice. We look at the tumor onset age. If we look at just normal regular BALB new T mice, again, first tumor appears at the age of 14 weeks, and it spreads out over a period of time. If you look at the BALB by DOF1 mice, yes, the tumor begins to appear early at 9 weeks. And by the time the mice are 14 weeks of age, more than half of them have developed spontaneous tumor. So DO pushed them forward. Now, did these mice, the BALB by PWK mice, capture that early tumor onset? And indeed, they did. By the time mice are 14 weeks of age, 100% of them have developed spontaneous tumor. So using these mice, we're able to capture that early tumor onset. And indeed, this is a genetic regulation of the tumor growth. And so, of course, by now we want very much to know what are the genes in these 3 QTLs that matters. And so we identify all the genes in these regions, and we hone in on those that have unique changes, missense or indel changes that are only found in PWK and Cas mice. And there are about 30 of those genes. So at this point, we got anxious and want to know if these 30 genes are also found in humans and whether or not they contribute to breast cancer. And so indeed, all but 1 genes have human homologue, and they distribute also in 3 clusters. And now they're in chromosome 2, X, and 6. And these 3 clusters are in similar orientation or sequence as in mice. So we interrogated 2 databases. One is the TCGA breast cancer database. The other one is an independent cancer data set that also shows survival and mRNA expression level. And we interrogated these 2 sets using these 30 genes. And more than 30% of those genes actually were found to be correlated with the breast cancer survival outcome. So now this panel of about a dozen genes become our target genes, and we would like to know, of course, how they function and how important they are. And perhaps we're going to need your help on, analyzing the genes, not just individually, but together. And we have many lessons to learn. But at this point, I'd just like to share the finding from one of the genes, that is ASCC3, which is associated with DNA repair. And in the TCGA database, it is shown that if a patient have high expression of ASCC versus those with low expression of ASCC3, the high expressors have worse outcome. And this finding was also validated in the independent breast cancer data set. So indeed, we have now a panel of genes that came out of the QTL analysis of DOF1 mice, and that is showing relevance in human breast cancer. And we would like to know how they work. So we just started to do this, and we took a BALB new T mice and its spontaneous tumor to look at which cells in the tumor actually express these genes. And of the clusters that are identified by the SURAT program, this is done by Jen Jacob, the junior faculty in my group. And you can see every cell cluster express this gene. It is apparently a very important gene and responsible for DNA repair. And the population that express the most is actually lymphocytes. And if you look at all the tumor cells, they all express it. So at the end of the day, who is calling the shot and making a difference would be of great interest. So I think we can say with reasonable confidence that this QTL analysis has given us information on which loci are regulating this HER2-neu induced memory tumor. And we have seen that PWK and Cas drove the more aggressive tumor, and we verified that using the BALB by PWK F1 new T mice. There are about 10 genes that evolved from this process that have unique variation in PWK and Cas, and turns out that they are the ones that also impact human breast cancer progression. So I think it's fair to say that DOF1 mice is an effective platform for discovering such regulatory genes. And last year, we also reported a paper showing using similar platform of mice expressing human HER2 in DOF1 mice, that a non-conventional MHC1 gene mediated the strong antibody response to HER2 vaccination. And this gene interact with the NK cell, and removal of NK cell also greatly reduced the response. So this platform is also very strong for identifying genes that regulate immunotherapy response. So I think I'm getting close to time, and I want to thank everybody who has contributed to this study. And it's a growing list, but those that are highlighted in blue are particularly important in this particular study. So thank you very much, and I'd be happy to answer questions.
Thank you, Wei-Zen. That's superb. You're really spot on 12 minutes. That's fantastic. So we've got time for a couple of questions.
Thank you very much.
So I don't see any hands. Maybe just.
Oh, I see Robert.
Oh, Robert's waving his real hand. That's right. I'm looking for.
I'm going to fight with Rob to ask questions before he does, but.
Okay,
I wish others would ask questions too, but I'll have one later on if nobody else does.
Bravo, this is great work. I'm sorry Kent Hunter isn't here to have heard your lovely talk, because he used this approach many years ago and became so successful that he quit doing genetics, which was a tragic loss. But this is definitely important work, and go for it. Where Liza Makowski at UT is using similar approach, but of course, because she's at UT, she's using BXDs. I just checked, completely non-overlapping QTLs that I can tell. So we're going to have a good time finding all of these germline variants that modulate cancer risk. Super important. So bravo.
Thank you very much. And I especially want to thank this community, we came to this study totally green, not knowing anything about how to do the analysis. And, you know, people in this community really guided us and foster our ability to do the analysis. Thank you. And I'm going to reach out to you again, you know, as we progress. Thank you. And Amelie?
Yes, then could you clarify, please, if the mice that you use all have the mutations, or do you also have mice that are wild type at the mutation? And are you looking at the interaction between the DO haplotypes and the NUT allele? Or are you just looking at mice that are all NUT mutants?
Okay, for the analysis, we only use those mice that did express NUT, even though the mice are kept as heads initially. So only one chromosome expressed NUT. Homozygous expression is actually embryonic lethal. So we cannot have homozygous. It's always heads. It's always only one chromosome having. And
but you could have wild types that do not have the mutation at all.
Correct. We have the siblings that are wild type that don't have it. We didn't do a whole lot of analysis on tumor onset, but in the previous paper, we have to compare the wild type immune response versus the one that expressed HER2. It's possible to do that. It's just, we breed a lot of animals. We have only so many that express HER2 and only so many that are female.
Okay, great. Thanks very much. We can move on to our next speaker, Anne Wells from the Jackson Laboratory.
Great. Okay, so the title of my talk is Natural Variation Alters Alzheimer's Related Gene Expression in DO Mice. So late onset Alzheimer's disease is the 6th leading cause of death in the United States as of 2018, with approximately 6.2 million Americans currently living with Alzheimer's. It's estimated that the cost of Alzheimer's and other dementias in 2021 is $355 billion, with the real cost actually being higher with a lot of unpaid help. Alzheimer's is considered a complex polygenetic disease, which is typically defined by cognitive impairment and beta amyloid plaques and tau tangles in the brain. However, currently, when we look at mice and try to study this, mouse models are currently poor as a study for this. So what we've done in this analysis is use diversity outbred mice, which many of you are familiar with, but using the heterogeneous population to mimic a human population more readily than an inbred strain. And what we did was we took 258 DO mice, both males and females, and they were just placed on a chow diet. So this is just simply baseline data. And these mice were genotyped on the Muga array. And then we took RNA-seq that from the hippocampal tissue of these mice, and the genes from the RNA sequencing data were grouped into 145 paracliques, which are unique clusters of genes that are connected through edges, and these are typically defined by correlation coefficients. And then on the flip side of this, to relate this back to humans, we have genes related to Alzheimer's disease from humans that were grouped into 30 human modules through AMP-AD. So to briefly go over a paraclique, so on the left side, we have a clique. And so here, this clique is a clique because every single node is connected through an edge. So all nodes are all connected through edges. But with a paraclique, we relax this idea a little bit, and we don't require every node to be connected to every other node by an edge. And so as long as a node is connected by one or two edges to another node, then that's considered a paraclique. And so in this way, we're relaxing our network to create a broader sense of what might be going on. And so for the human modules, we have 3 cohorts. We have RosMap, Mount Sinai Brain Bank, and Mayo Clinic. And these all make up 7 brain regions across the 3 different cohorts. And so the human data has been aggregated into these 30 modules, and AMP-AD stands for Accelerating Medicine's Partnership for Alzheimer's Disease. And so these modules here were derived from multiple clustering algorithms. And within each of these modules are genes that have been enriched in differentially expressed genes involved in AD versus control. So this has been a case control study for the humans. And so for the methods of what I'm presenting today, what I originally did was regress out the cis effects from the rank C normalized data. And so this is in regards to the DO hippocampal data. I performed the EQTL for all the genes identified in the transcriptome, because I should specify here, the transcriptome has more genes than are in the paracliques. So I first started with the whole data set before I went down to what genes were present in the paracliques. So here I identified cis and trans EQTL, regressed out the cis effects, performed EQTL with the residuals, and then independently assessed potential candidate genes identified through the significant LOD scores from the paracliques. And so to look at QTL for the paracliques, what I did was I created eigengenes using singular value decomposition. And this was just to summarize the genes in each paraclique, and then performed QTL analysis on those summary eigengenes to identify potential candidate genes and allele effects. And then I compared these genes within the paracliques and the AMP-AD to identify significant overlaps using Jaccard index. So just looking at the total RNA first here, just to give a sense of what's going on in the data, we can see that there's a lot of distal and local effects with some potential trans bands at 5 and 7. And then when just looking at the genes that are inside the paracliques, this is sort of what we're looking at here. So there are probably roughly about 1,900 genes in the paracliques versus about like 17,000 in the total. And this is just through the graph algorithm that creates the paracliques. It really hones in on what's most correlated. And so here again, you can see though that there's still local and distal effects. And so then looking at the transband plot, we again can see that there are some transbands at 5, 7, and 17, looking at all of the RNA data. So when we're looking at how to compare the paracliques to the AMP-AD modules and assessing this mouse versus human, we see that there's an order of magnitude of difference here. So I just wanted to point that out just to be transparent, that the largest paraclique contains 407 genes, and paracliques are non-repeatable. So once a gene is in one paraclique, it won't be in another. And so you can see the largest paraclique is really big compared to the last paraclique. And this is done by design, again, with this algorithm. And so the smallest paraclique contains about 5 genes, whereas the modules are sort of all over the place, but all relatively within 100 to several thousand. And so when we look here at the Jaccard overlap, so here, again, I have the plot in the corner of our original AMP-AD modules, and I have ordered the AMP-AD modules in the same order that you see over here for consistency. And then up top, I have put the classifications of which type of functional classification each module has been given. And so here you'll see that although the legend is pretty small, so we're going from 0 to 0.005, this has to do with the fact that the overlaps are small because of that order of magnitude of difference. And then these numbers on the right are the paraclique numbers. And these, instead of doing all 145, what I did was I only listed the ones here in this plot that had a significant QTL to start so that you could see better what's going on. And so what we see here mostly is that there's significant overlap with the modules in the AMP-AD modules with the paracliques for astrocyte and for neuronal module classes. So then starting to look at what's going on inside of these paracliques and potential candidate genes, I started with the 28 paracliques that were significant for some QTL. Then I looked at the SNP associations and looked for anything that had a potential neuronal effect, and that whittled me down to 11. And then I looked at SNP associations that were identified in a database called Agora, which is where all of the Alzheimer's researchers can go in and they can put their data in and start to aggregate information and determine nominated targets for downstream analysis or for potential therapeutics for Alzheimer's. And so that really whittled me down to 4 paracliques. And so I'm only going to talk about one here because we don't have a lot of time, but this will give you a sense of sort of how this is working. And so here, looking at paraclique 79, we saw that CAST had the strongest effect on paraclique 79, but we also see that PWK and AJ here also have an effect on this one. And looking at this paraclique, we saw there were 16 SNP associations and that this overlapped with the astrocyte AMP-AD module. Although it wasn't statistically significant, it trended towards significance. And so when I looked at the SNP associations, I saw SIRF1 as one of the genes that was affected. And looking at the EQTL for SIRF1, I saw that PWK also had the strongest effect here, and that SIRF1 plays a role in AD, but defects are actually known to cause Lee syndrome and Charcot-Marie tooth disease, and that mutations in this gene affect calcium uptake by mitochondria in the neurons. And then to see if there were potentially any genes mediating SIRF1 and paraclique 79, we saw that looking at global and local, we saw that there was a local mediator, SLC1A2. And looking at the QTL effects of SLC1A2, we again saw that PWK had the strongest effect on this gene, and that this gene is a nominated target in Agora and has been shown to be decreased in Alzheimer's disease brains, especially in hippocampal tissue from late stage Alzheimer's patients. And that reduced expression of this actually is a part of the procession from mild cognitive impairment to Alzheimer's disease. So before Alzheimer's disease, there's this other thing called mild cognitive impairment, where someone is just starting to forget mild things, but it's not progressed enough to be diagnosed as Alzheimer's disease. So in summary, paraclique genes were largely overlapped with the neuronal and astrocyte AMP-AD modules. We saw that the QTL analysis identified genes in the DO mice that are present in the AMP-AD modules, and that QTL mapping suggests that LC1A2 may act as a master regulator for genes in paraclique 79. So some future things I'm working on in a list of many is to assess motif effects, and then also to compare some GWAS hits from the AMP-AD human modules with the SNP associations from the paraclique QTL. And with that, I would like to thank lots of people. This has been a big project with Model AD and several labs across campuses and universities. And with that, if you have any questions.
That's great. Thanks very much. And we have time for some questions for Anne. I don't see any hands, but if you do, we can just shout. Okay, and I've got a question. You had your microglia in there. Did something come up for the microglia by any chance?
You know, I don't know about specifics for microglia because everything was sort of bulk. One thing that we could look into is trying to see if we could use other databases to sort of hone in on microglia. We certainly have others in the lab, but I can't say anything super specific.
Great.
I think using paracliques is so clever. I just like this method quite a bit better than using WGCNA. It's not exclusive. It's quite a bit more flexible and frankly, more comprehensible than the raising to an exponent of 9 or 10 or 7 to clean up your correlation coefficients. So I love it.
Thank you.
Okay, great. Thanks so much, Anne. And we can now move on to our next speaker, who's Jacob Beierle from Boston University. Jacob.
All right, let me take it away. So I'm going to talk about efforts that we have conducted in the Laboratory of Addiction Genetics under Cameron Bryant to characterize some differences in oxycodone behaviors and really pharmacokinetics. But before I get into that, because I have a bad habit of running long, I'd just like to thank everybody ahead of time. Everybody from the Bryant Lab at BU, particularly Emily, Anya, and Will, who's taking over the project when I'm gone. Stan Goldstein did a lot of the proteomics stuff, and then we have Marty Ferris giving us whole genome sequences for some of these strains for the BYJs. Dr. Moody leading the metabolomic mass spec out in University of Utah, and Dr. Peltz. We have a collaboration that this data came out of with him. So big thanks to everybody there. So like I said, I'm going to talk about opioid-related traits, and I don't want to belabor the point because I think everybody understands that the opioid epidemic is a real problem in the United States, but the statistic that I find encapsulates it very well is that in 2018, more Americans died from opioid-related overdose deaths than from auto fatalities. I think that's pretty staggering. And in the current COVID environment, this crisis is only getting worse. So we know that opioid use disorder is heritable from twin and familial studies, but GWASs and other human studies haven't really fully explained that variability. And so, like many of you, that's the question I'm trying to answer. I think we all understand why animal models are useful. So before I dive into the behavior, we need to talk about the actual metabolism of oxycodone. Oxycodone is a semi-synthetic opioid that is prescribed primarily because of its high oral availability, bioavailability. It's primarily metabolized through phase 1 metabolism enzymes, the CYP enzymes, into the inactive noroxycodone through CYP3A4 in humans and into the active oxymorphone by CYP2D6. Oxymorphone only represents about 10% of the overall metabolites created from oxycodone metabolism, but it is much more potent at the mu opioid receptor, about 8 times as potent. And noroxycodone represents about 70% of the overall metabolism, but is not active at the mu opioid receptor, very lowly binding. So that's the context of oxycodone. Let's talk about the behavior. So we used substrains of BALB-C mice, specifically BALB-C BYJ and BALB-CJ. And we tested them for this high throughput assay for drug preference called conditioned place preference. Essentially, over 4 days, you train the mouse to associate drug with one context and saline with another. And the difference between their drive to spend time on the drug paired side from pre to post test is an operationalized method or an operationalized measure of reward. Excuse me. What we find is after training, when we test these animals for their preference and we only administer them saline, so in a drug-free state, there's no real difference in these substrains, and you'll actually see very small learning. This is a low 1.25 mg per kg dose. But when we prime animals tested with oxycodone, when we prime animals trained with oxycodone before testing them for preference on day 9, we see a huge difference, or a very large difference, I should say, between the BYJ and the J animals, which is suggesting that there is something about this dose of oxycodone, this introspective cue, that's driving the J animals to spend more time on the drug paired side. When we break this down by sex, what you see is the difference between the female animals is much larger than the difference between the male animals. And this parental strain effect is largely being driven by the increase in J females and this decrease in the BYJ females. So that's all well and good, but we're giving these animals drugs, and so we had to consider pharmacokinetics. At the recommendation of Camron, we pulled about 16 brains for this study just as a last-minute effort, and they're probably the most important 16 animals of my PhD right now. What we saw was, overall, there's an increased availability of oxycodone in CJ animals. But more striking, I think, is the fact that the metabolites in CJ females are increased, and they are the only group within these 4 groups that is increased. This parallels our CPP data nicely and suggests that maybe increased oxymorphone is driving increased preference, and that this is not an introspective learning phenotype, but instead a phenotype driven by pharmacokinetics that alter the salience of that priming cue. So as we all know, the next step is to make an F2 cross. We use the mini Muga microarray to genotype these substrains. It's perfectly designed for that. It's fantastic. We also added another marker, which I'll talk about later. But the real advantage that I want to hit on really briefly, the real advantage to using these BALB-C substrains is because they are so closely related, we greatly reduce the polymorphic density between the two. So these mice differ by about 8,500 SNPs and indels based on short read sequencing. There's much more dramatic structural variation going on and copy number variation, but that's harder to track without long read sequencing. We do have some evidence for that in our EQTL data. But in addition to offsetting offsetting the large interval size with reduced polymorphic density, you also increase your chance that your change in phenotype is being mediated by a single causal genetic factor, and also by extension reduces the chance for gene by gene interactions on these backgrounds. So when we made our F2 population and pulled brains after injecting with oxycodone, what we found is a single region on chromosome 15 that is significantly associated with changes in whole brain concentrations of oxymorphone, that potent metabolite, but not oxycodone or noroxycodone. If we zoom in on this peak and just look at chromosome 15, what you can see is, in red, the females are largely driving this effect. And the males really only show a slight little bump here, right? This is not significant if you run this by themselves. And when you look at the effect plot at our peak associated marker right at 23.8 centimorgan, you can see that we really nicely recapitulate our parental strain data. We don't see any real differences between the CJ males and the BYJ groups. And we see a real stark increase in the oxymorphone concentration within the female CJ brains. I mean, within the female group, this locus explains 32% of the overall variance in the phenotype. So really nice that we recapitulated this. So we paired this analysis, right? We now have to go select candidate genes. And the easiest way to do that is to look at expression data and do EQTL mapping, so that we can understand what genes within this interval are differentially expressed and select them as candidates for validation. As my talk title kind of gives away, when we did EQTL analysis in these animals, we only identified a single significant EQTL in both striatal and hippocampal RNA samples. I should mention this is a cohort of 64 animals, so it's a very large sample size. And we only have one gene significantly differentially regulated in this region, in both the striatum and the hippocampus, based on our FDR values, and that is ZHX2, which, yeah, we'll get into that in a second. So what's nice, and the reason that we added this middle marker right here is our expression data, our EQTL data, is most significantly associated with the markers that are surrounding ZHX2. This additional marker we used to move our peak to the left, and I'm still working on genotyping all of the EQTL animals, but I suspect that this will be the most significant marker based on its proximity to ZHX2. We also, in addition to EQTL analysis in F2 animals, did whole brain proteomics in parental substrains. So what you're looking at is a volcano plot demonstrating the change in expression. On the right are genes that are proteins that are highly expressed or more highly expressed in the J animals, and on the left you have the downregulated proteins. This was collected right after our final day of CPP testing, because that was what we were initially interested in. As you can see, ZHX2, which corroborates our expression QTL data, is downregulated in the, I'm sorry, I've switched this up, I apologize. The BYJs show a much higher increase than the CJ animals. So it's nice to have that expression QTL data demonstrating that as well. So what is ZHX2 and why is it important? Well, some of us know the answer. Jake Lucis has done work on this before, describing ZHX2's effects on liver fat and lipid metabolism in general between BALB/C substrains. But long story short, ZHX2 is a zinc finger and homeobox protein that is a transcriptional repressor. And the consequences of hypomorphic ZHX2 are alterations in metabolism, right? Fat is endobiotic that's metabolized through the liver as well as xenobiotics. But most importantly, there is a demonstrated sex-specific modulation of cytochrome P450 genes in the liver. And that is primarily, and our difference in expression in these animals is driven primarily by an inserted retroviral element within this gene that causes inappropriate splicing and hypoexpression of the functional protein. So to wrap this up, to summarize, we think that these BALB/C/J animals are showing increased metabolism of oxycodone or decreased metabolism of oxymorphone. And that's causing increased brain concentrations of oxymorphone and leading to strain-specific addiction behaviors, wherein CJ females show more addictive properties in a CPP paradigm. So the next step for this project is to take the CJ animals and repair the 6.2 kilobase insert within ZHX2, repair their expression, and demonstrate that that's sufficient to rescue the differences in metabolism that we see. And so with that, I would be happy to take any questions.
That's great. Thanks very much, Jacob. Any questions for Jacob? Jacob, can you remind me, in your human data on the opiates, is there a sex-specific effect?
Is there a sex-specific effect? Well, so the human data is challenging. So I don't know of a demonstrated sex-specific difference in any phenotypic outcome for oxycodone. To answer your question.
Maybe this was the question, but I didn't get, and perhaps you said and I missed it, why is this a female-specific phenotypic effect? What, it didn't look like there was a sex specificity in the expression, or did I miss that?
So in ZHX2 expression, there is not a sex-specific effect, right, but there is a sex-specific effect of cytochrome P450 genes when you knock out ZHX2 within the liver of adult mice. So ZHX2 hypoexpression or an animal that is hypomorphic for ZHX2 will have sex-specific dysregulation of cytochrome P450 genes. And we suspect that that is the causal mechanism, or at least some transcriptional change from ZHX2 is the causal mechanism. Like I said, it could be cytochrome P450 metabolism and oxycodone, or it could be UGTs and oxymorphone.
So in your gene expression data, though, did you see that or not?
In my gene expression data, did I see? Oh, so, yes.
You got RNA-seq data, so wouldn't you expect to have seen that, or were you in the wrong tissue to see it?
So here's what we did see. We did not take, let me answer your question really quickly. Within the brain, we see differential expression of CYP2D11, right, which is possible, and we see differences, large sex-specific differences in this expression, right? We also see when we look at liver data, sex-specific differential expression of UGT genes. So to answer your question, it's hard for us to look at the proteomic brain data because it's in parental strains and kind of tease apart what effects EHX2 is having downstream. And so we can only really look at this liver data, and based on the liver data is also from parental strains. So we're kind of trapped. We can't correlate ZHX2 genotype with differential expression because we don't necessarily have high expression of cytochrome P450 genes in the brain, and we didn't really have good enough depth of sequencing. I'm sorry, I realize what you're asking now. And we didn't have good enough depth of sequencing.
The thing you said is the key point. Yeah.
To capture the cytochrome P450 expression.
Okay.
And this is the only gene that comes up within the brain, and it's proteomics.
Makes sense. One last question from Aldens.
Yeah, Jake.
Jake.
Unmute yourself.
Yeah, I'd love to hear from you, Dr. Lucis.
Oh, but Jake, you're still muted.
Thank you so much. We made a congenic where we took the BALB-C allele and put it on the Beierle allele of BALB-C, and we've noticed some behavioral differences, or people in the lab have, not me actually personally, and sort of like startle response effects, and we just didn't know what to do with it. We tried to see if it was we could replicate it, and it doesn't replicate very well. But you know, it's kind of interesting in light of your studies. You know, in the context of atherosclerosis, ZHX2 affects apoptosis of macrophages. That a very significant effect in atherosclerosis. The allele affects. Oh, shoot. Too many things going on here. So anyway, I don't know if in any way, Jacob, that would relate to your findings, but that was very interesting. Thank you so much.
Yeah, no, thank you for your input. That's very interesting to consider.
Great. Okay. In which case, let's move on to the last speaker of this session, Cameron Bryant.
Not hearing anything, so I'll assume so. Everyone can see this?
Yeah, good. Yeah.
Great. All right, so I'm going to take the same approach as Jake. I'm going to first thank everyone, including Jake, who generated most of this data, Emily Yao, who got the QTL analysis started in our lab with these with this cross, and various collaborators, including Andrew Mealy, who's responsible for the proteomics work, his student Stan Goldstein, who's shown here. Recent collaborators that I've recruited to look at BALB-C pain phenotypes, including Dr. Aaron Young at KU Medical Center, as well as Ahmad Damaj at Virginia Commonwealth. This work was spawned from a U01 that's funded by Gary Peltz. We snuck in these substrains to see if we could find a difference. The idea of, you know, perhaps getting a new U01 from this, and luckily it worked. And so I have Gary to thank for that. And Marty Ferris, who has provided the whole genome sequencing data and continues to advise us on the DNA side of things. And then last but not least, funding from NIH, NIDA, NIGMS, and Burroughs Wellcome. Okay, so today I'm going to tell you about hot plate sensitivity, which is shown down here. So here, it's the measure of thermal nociception. We put the mice on a hot plate, measure how long it takes before they flick their hind paw. So I want to point out to the pain enthusiasts that this is not technically a pain response, it's a nociceptive response. And so what we mean by that is you apply acute stimulus, in this case thermal, which is an unpleasant, noxious stimulus. You have activation of nociceptors. So nociceptors can be thermal, mechanical, chemical, or polymodal. And then you have the transmission of nociception from these peripheral nociceptors. And these are the dorsal root ganglia here, so cell bodies in the periphery, and they have axons that synapse into the dorsal horn of the spinal cord. You then have secondary transmission from the spinal cord up to the thalamus in the brain, and then ultimately tertiary in the cortex where pain is experienced. And like I said, the response we're looking at is withdrawal from the heat source, in this case, the hind paw of the mouse. Okay, so why study acute nociception? This is definitely a difficult question to answer for the pain audiences. The short answer, the practical answer is it's very quick to study. This is kind of a side project from the oxycodone study. And so we decided we would just include it in our phenotypic battery. And luckily, we got a QTL for it. But it does have evolutionary importance. So there's this phenomenon, congenital insensitivity to pain. And these individuals do not experience any pain symptoms. And usually they die much earlier on than they should because of this, through injuries and so on. There was a recent study that came out a few years ago with the FA-out gene, which is the pseudogene. It's involved in cannabinoid signaling, and this individual had no pain and actually very, very reduced fear and anxiety as well. And she's actually still alive. So acute pain can actually become persistent if not treated in some fashion. And so the idea is that by studying the genetic mechanisms of acute pain, we can perhaps identify ways to prevent the transition from acute to chronic pain by relieving it, and then ultimately used in therapeutic development. Okay, so this is by all means not a conclusive list of what all is known about the genetics of acute pain, but these are just some major examples. So for instance, this gene Hydin was identified from the Chesler group using DO mice. We identified a candidate gene in a reduced complexity cross between B6 substrains a few years ago. And the most obvious candidate gene in this case was a ryanidine 1 receptor based on not only eQTL, but also historical literature, number of polymorphisms, deleterious polymorphisms, and so on. Jeff Mogul, several years ago, identified the candidate gene OPRD1 for her hot plate sensitivity as well, which is interesting because OPRD1 now is strongly implicated in chronic pain. We see adaptations in the delta receptor in surface localization in chronic pain models. So this perhaps might be an example where studying acute pain can lead to discoveries related to chronic pain. And then finally, in the tail withdrawal assay, which is another assay of thermal nociception, this gene where you stick their tail in hot water and measure how long it takes for them to flick it out. This gene TERP1 was identified on chromosome 4 using AXB, BXE recombinant congenic strains. Okay, so Jake went over reduced complexity crosses. So I'll try to minimize repeating here. They differ by about 8,000 variants, including SNPs and indels. The hot plate assay is our primary phenotype here, 53.5 degrees Celsius. And you can see a very robust difference between BALB-CJ and BALB-BYJ. Latency drops from 30 seconds down to about 20 seconds. And so we have this marker panel from the mini Muga that we used for QTL mapping. We have 284 F2 mice. We used RQTL. We included sex in prior treatment as additive covariates. So some of these animals, approximately half of them, had previously received a few injections of oxycodone. Turns out this doesn't matter. You can actually map it without those mice in the saline controls just as easily, but we included them anyway in the model. And then finally, well, in addition to proteomic analysis, we did cis-eQTL analysis in 64 F2 mice, which in hindsight is probably overkill, but we can be very confident in our eQTLs that we have identified. Okay, so this is probably the most striking, beautiful F2 QTL that I've seen from my lab so far, the LAS score of 10. And what's really striking is that we've really accounted for pretty much all of the variants. I mean, if you look at the drop from 30 to 20 seconds, we see almost exactly the same drop at this locus here, the peak associated marker, which explains 18% of the phenotypic variants. And so we then looked at genes with predicted highly deleterious polymorphisms within this interval, which are all shown here. So we excluded upstream, downstream, intergenic, and intronic that were non-splice, just to help whittle down our number of candidates. And this gene, I'll just point out here, H2-AFI, is an interesting candidate gene that I'll show you why in a moment. Actually, there's nothing known about H2-AFI and pain sensitivity, but nevertheless, we've found it to be a compelling gene based on our subsequent analyses here. And this gene is located right near the peak of the QTL at 56 megabase, approximately 30 centimorgans. When we do cis expression QTL analysis, this is from striatal tissue. We see that H2-AFI is the top cis QTL within this region, easily surviving the FDR cutoff. And so at this point, this is our highest priority candidate gene. Actually, none of the other genes in this interval technically survived the FDR cutoff. And so whole brain proteomic analysis, as Jake went over earlier, further supports the candidacy of this gene, H2-AFI. You can see that there's much higher expression here in the J versus the BYJ. So approximately 4-fold increase expression. This is log 2-fold change on the x-axis. And look, Abe, there's GLO1 there. It's our positive control. So I was happy to see that. It's known that the J's have a duplication, I think. Or is it the delete? I think it's the BALB J's have a duplication within the GLO1 gene. And here we're just plotting normalized intensity on the right. You can see the much higher expression protein in the J versus the BYJ of H2-AFI. And like I said, there's nothing known about this gene in terms of pain sensitivity. It's a histone. It replaces 2A histones in the nucleosomes to repress transcription. It's involved in X chromosome inactivation. Trying to see how much time I have left here. Okay. And a few other things I've shown here. I won't read through everything, but the point is there's really nothing known about its potential role in pain sensitivity. And just because I have it in the title, I figured I'd throw this in, but I realize I'm running out of time here, so I'll just go over this briefly. There is historical literature indicating brain morphological differences between BALB-C substrains, and probably the most gross phenotypic measure is brain weight. So again, we wanted to be able to take as many easy phenotypes as we could when we were dissecting these mice. So we did brain weight QTLs on I guess it was on 256 of them. And you can see that there are 2 genome-wide significant QTLs. By far the biggest one here is on chromosome 5. And we also found one on chromosome 10, although in the predicted opposite direction of the parental strains. And again, what you'll see if you plot the brain weights, you have about 0.46 dropping down to about 0.41. This is grams. And you can see the effect plot almost exactly recapitulates it in the F2 cross at the peak associated marker on chromosome 5. So we think this is the major locus underlying brain weight differences in these strains. Just to point out, these are again, the most highly deleterious mutations within this QTL region, occurs in these genes. Interestingly enough, though, we didn't find any cis eQTLs that survived our FDR within this region. And none of these genes that we did find that are even suggestive overlapped with these genes. So at this point, we didn't really have any compelling candidate genes. But we go back to our trusty old proteomic data set. And what we find is a very large increase in expression of ACADs in the J strain versus the BYJ strain. And Jake was able to do some digging through the literature. And what we found is just like the ZHX2, there is a private deletion, but in this case, it's within the BYJ strain within this gene, which interestingly does not appear to affect RNA levels. Otherwise, we would have detected it. And I think that's been shown in the literature too. But at the protein level, we see a very large difference in expression. And so we think that this structural variant is actually responsible for this difference in expression, and potentially a difference in brain weight. This gene, yeah, I don't have time to go over this gene, but there's some interesting prior literature that suggests that this is a very high priority candidate gene. So to conclude, systems genetic analysis in the BALB-C substrains, reduced complexity cross identified H2-AFI, the candidate gene for thermal nociception, and ACADs for brain weight. And so we've shown that expanding RCCs to BALB substrain, this approach reiterates the promise of the approach for rapid and efficient complex trait analysis. And finally, these studies highlight the need for long-read sequencing for structural variant identification, as well as the needs for proteomics and systems genetic analysis. So, ZHX2 and ACADS, without the historical literature, we wouldn't have been able to implicate them. And furthermore, with proteomics, we were able to identify ACADS as a candidate gene, which we couldn't have done through EQTL analysis alone. And so with that, on the East Coast, it's almost lunchtime. I will take questions if we have time.